Hello and welcome to our final video in this unit. Today what we're going to be discussing is protein synthesis, or rather the process of going from DNA to RNA and ultimately making a protein from that strand of RNA. Now this is going to be probably a little bit longer than some of the other videos. We were going to be tossing a lot of information at you, but take it one step at a time and I'm sure you'll make it out just fine. Well, let's get started. So the first step in going from a strand of DNA to a protein is called transcription, or making a copy. In this case, it's the process of making a mRNA strand from a single strand of DNA. Now, the nitrogen bases in RNA always will bond to their complement on the DNA strand. Now these follow rules very similar to Chargaff's rule, except in this case, A is going to go to U, and G and C are going to swap. So what you're going to go ahead and see here is let's have DNA over here, and let's have RNA over here. So if we have A, T, G, and C, if we had A on the DNA, A would pair up with U on the RNA. If we had a T on the DNA, that would pair up with A on the RNA. If we had a G on the DNA, sorry about that, that would not go to a G, that would go to a C, and C would then go ahead and correspond with G. So AU, TA, GC, CG. This will make a bit more sense as we progress. So the steps in transcription. The first step is the enzyme RNA polymerase is going to go through and unzip the complementary strands of DNA into two single strands, as you can see currently right down here. So the RNA polymerase is going ahead and unzipping the strands of DNA. From there, the RNA nucleotides bond to a single strand of DNA. And then once that's said and done, the mRNA is released and the two DNA strands rezip. So what we have here is we've got our RNA right here, this red strand, and we have our DNA up top. And as you can see, it's going down and making a copy of one of the strands by pairing up with its complementary base pair. So here we have U and A pairing up. Here we had a T on the DNA, so that goes to an A. We had a C on the DNA, so that went to a G. A G on the DNA went to a C. An A on the DNA went to a U, and so on and so forth. And this would continue until it has an exact copy of one of the strands that it needs. So mRNA processing. Now enzymes can go ahead and remove or cut out introns, or parts of the DNA that they don't need. So let's say, for example, in this case, we didn't want to either have these two G's or this U. So what it can do is go ahead and remove those. So enzymes can remove introns and cut them out. We don't want them because introns interrupt. What we want are exons. What's left behind are known as exons because they are what are eventually expressed or translated to make our proteins. So like we said, this was our original copied strand, this was our original strand of DNA. We didn't want this part or this part. So we go ahead, create the mRNA, and once we have these copied, we just go ahead and remove them. So our eventual RNA strand that we have at the end, instead of having 1, 2, 3, 4, 5 parts, only has 3. So all this is trying to say is the RNA can go ahead and modify it as it sees fit to make sure that it has the right stuff. So what I want you to do here is remember Chargaff's rule, except this time we're going from DNA to RNA. So remember, DNA, A, T, G, C, RNA, A goes to U, T would go to A, G to C, and C to G. So I'd like you to go ahead and pause it right now and work your way through this. And then go ahead and unpause it when you're done and check your answer. Okay, so let's see how you did. So TAC would go to AUG, TCG would go to AGC, TCC would go to AGG, ATA would go to UAU, GGC would go to CCG, and ATC would go to UAG. Now, if you've noticed, all of these have been in sets of 3. So at any given time, we had 3 bases, or 3 letters, in one sequence. There's a reason for this, and let's talk about what that reason is. These are what we call codons. Now, a codon is any group of 3 bases on an mRNA strand. There are a possible 64 different codons that encode for 20 different amino acids and a start-stop signal, so telling it when to start and when to stop. And this genetic code is universal amongst all organisms. So, if you take a look, that means we have 64 codons but only 20 amino acids. So this means that multiple amino acids will have a similar codon combination, which we'll discuss here in a little bit. But up top we have GCU, that's our first codon, ACG our second, GAG our third, and so on and so forth. So this right here is what we call a codon wheel. And a codon wheel is how you go ahead and go from a strand of codons, or mRNA, to your amino acids and ultimately figure out what is going to be part of your protein. So we have our strand of mRNA here. What I always do is to go ahead and separate these into the basic codons, or sets of 3, just because it's a little bit easier to go ahead and read. So how we go ahead and use this is as follows. So let's go ahead and do our first codon, GCA. So we start off with the letter G on the inside. So you always start in the middle and work your way out. So GCA, start off at G, go to C, and go to A. That gives us the amino acid alanine. If we go ACG, we start off with A, move to C, and then move to G. That gives us the amino acid threonine. UUG, start off with U, move to U, then to G, gives us the amino acid leucine. CUA, start off with C, move to U, move to A, also gives us leucine. And let's do one more, like CUG. So, start off with C, we go to U, then we go to G, which also gives us leucine. So what this means here is multiple codons can give you the same amino acid, which is why we have 64 different codon combinations, but we only have 20 amino acids. So let's talk about that process, going from mRNA to your amino acids. This is called translation. So translation is the process of using RNA to assemble amino acids into a protein. And here are the steps. So let's go through one at a time. So the first step is the mRNA, now that it's copied, leaves the nucleus into the cytoplasm and finds a ribosome. So the rRNA, the ribosome, will then go ahead and read off the codons one at a time. So it reads off the sets of 3 bases. Once it goes ahead and reads those off, it sends a signal for the correct tRNA carrying the corresponding amino acid to the ribosome. Now once the tRNA gets there, this thing called the anticodon on the tRNA then matches with the codon to make sure the right amino acid was brought. So all that's doing is it's going ahead and it's having the base pairs of whatever is on the codon. So let's say on the codon we had U. On the anticodon, it would have U, A. And the whole reason that's there is just to go ahead and make sure it brought the right amino acid. Now, once it goes ahead and matches up with the correct amino acid, the amino acid is let go by the tRNA. And the process continues. It kind of reads it down like a tape. So it just goes through codon by codon, sets of 3 by sets of 3, until ultimately it forms your polypeptide, or your string of amino acids. This will continue until it goes ahead and reaches what is known as the stop codon. So the stop codon in the mRNA tells the process to end, and the new protein, or the string of amino acids, is then let go. So the way this works, like we said, here we can see the mRNA going ahead and making its way out of the nucleus. And it continues this until it goes through and finds a ribosome. So here, our ribosome is going ahead and reading off our codons on the mRNA. And it's reading these off one at a time. So here, we have our first codon, AUG. So, in comes our tRNA. So we have our tRNA here. With it, we have two different things. We have our amino acid up top, and we have our anticodon. So as you can see, all the anticodon's doing is going ahead and just matching things up to make sure, okay, yes, I did indeed bring the right amino acid. Because on the mRNA we had AUG, and on the tRNA we had the anticodon UAC. U going with A going with U, and C going with G. So it goes through and reads off codon by codon, continually going ahead and bringing in new tRNAs along with their amino acids. So this continues until we eventually get a string of amino acids, and these are held together with peptide bonds, creating what we call a polypeptide. So this will continue until the last codon has been read, and once that's said and done, we will have a completed protein, which will then go ahead and move off to wherever it needs to be. So, real quick, I'd like you to go ahead and once again pause the video and use your codon wheel to find the correct amino acids for the following codons. So pause it now, and then come back to check your answers. Alright, let's go ahead and see how you did. So AUG gives us methionine, or MET. CAU gives you histidine. AGC, serine, and CUA, leucine. So, now this is kind of corny, I understand that, but it might help you remember the steps. So remember, we start off with DNA, then we go through transcription to get mRNA. Then after we have our mRNA, we go through translation and get our protein. So the way I remember this is with dude, teach me to paint. So, dude corresponds with DNA, that's where we start. Teach, transcription, me, mRNA, to, translation, paint, protein. So dude, teach me to paint. I know it's kind of out there, but it might help you remember the entire process of protein synthesis and how it starts and ultimately how it ends. So, with that, I know we covered a lot today, and this is one of those that I really want you to go ahead and practice to make sure you have it down. This is going to be a huge part of this unit. Now, if you have any questions on this entire process, I highly encourage you to go ahead and talk to your instructor. They're going to go ahead and help point you in the right direction and help you with any confusion you may have. Now, if you got it, that's great. And if you don't get it, don't hesitate to ask for help. Anyway, that's enough of me talking over here. So, thank you very much for your time. And until the next video, you take care, and I'll see you then.